restriction enzymes, or restriction endonucleases, are sequence-specific DNA cutters, and so they recognize specific sequences in DNA and cut them. The name restriction comes from the use bacteria use them as part of a restriction modification system, and whereby they modify their own DNA with methylation and then recognize these sequences in foreign DNA from bacteriophages or phages, which are these viruses that infect bacteria. So they're able to restrict or limit the activities of these phages, but for us, the use of these enzymes is pretty limitless, which is why there are so many that we can buy commercially. And these all like recognize different and cut different sequences and that sort of thing. And so what do we use them for? All sorts of things. A key use is restriction enzyme cloning. So basically we take DNA from one place and we cut it out with restriction enzymes, and we cut out another piece of DNA with restriction enzymes, and then we stitch them together. We can also then use restriction enzymes to check and see if that cloning worked by cutting sequences like in between them and that sort of thing to see, and then looking to see how big the pieces are they as big as we would expect. We can see, speaking of size of pieces, we can take advantage of natural variations in DNA sequences and sequence length, like polymorphisms. There's this thing called like restriction fragment length polymorphisms. Basically, people have different length sequences, then you can use this to like track genetics and stuff. It was more used in the past than in the current day, where we have more sequencing techniques. But all of these various uses for these restriction enzymes, they're super duper useful, but some of the terminology can be kind of confusing. Also, the fact that the restriction enzymes, because the bacteria normally use them as part of this like restriction modification system where they're methylating their own DNA, so they're modifying their DNA, and also like trying to avoid using the sequences that they then attack, but they're modifying their DNA and then they're attacking the foreign DNA. But sometimes the phages get smart and then the bacteria like attack back and that sort of thing, and so you get these cases where you can have methylation that's actually interfering with the restriction enzyme you're trying to use. So there's some different technical notes that we'll get into about potential conflicts that you might run into when you are trying to do this sort of thing. We'll also get into some of the weird buffer names like YUF3, 3.1, R 3.1, cut smart, all of these different buffers. Things like star activity, various things that have to do with how specific an enzyme is acting and how well it acts in different buffers and how they're optimizing buffers and stuff. Basically, NEB, New England Biolabs, I do not work for them. I got this poster from a conference, and basically NEB has like every restriction enzyme you could ever want, and they're kind of like the go-to place for restriction enzymes. And so I'll show you also on their website and a couple of other sites where you can actually design like what enzymes you want to use, introduce sites if you need to do that, like silent sites, various things like that, as well as some technical notes about using them. But first, let's start off in talking about where these are, because although they're super duper useful for us, they're actually super duper useful for bacteria, and they're so useful about bacteria that they actually like help these bacteria survive. And so it's important to understand where they're actually coming from, and because the bacteria didn't make them for us, they made them for themselves. So let's dive in. So first, a little terminology. So the term endonuclease, that's basically referring to the fact that it is cutting in the middle of DNA. And so if it was, we said exo, that would mean it was chewing from the ends, but an endonuclease is going to cut somewhere in the middle. And what's really special about restriction endonucleases is that they cut DNA in at very specific sequences. So they're not just like randomly chewing up DNA. And these sequences that they recognize are often palindromic. So basically think like kayak, the same if read in both directions, but in this case, it's the same on read on both strands. And so DNA strands, two strands, and they're complementary. So G pairs with C, A pairs with T. And these sequences that these restriction enzymes recognize are often palindromic. So they're read like the same on both strands, because when you think about reading, you're talking about like 5' to 3'. And the restriction enzymes that we typically use are these like type 2 restriction enzymes. There are some restriction enzymes that like actually recognize, they cut outside of the sequence they recognize, and there's some weird types of restriction enzymes. But the type that we
we typically use in the lab are things where they're recognizing this specific sequence and then they're cutting inside of it. And typically they start acting as like a dimer. So you have one copy of the enzyme binding to each strand, and then they're cutting each of these strands. They can cut, some of them cut like a straight cut, and these will produce blunt ends, whereas others cut in this staggered format, and these produce sticky ends. And these sticky ends are really useful for when we're trying to do things like restriction cloning, where we can cut pieces together and then stitch them together with the DNA ligase. And these, you often use a combination of restriction enzymes, and you can even use different restriction enzymes that recognize slightly different sequences, but they make the same like overhangs that you can then stitch together. So what do all of these names? So like BAM-H1, EcoR1. So I was, those, first of all, those like ones, and sometimes you'll see like threes and fives. So this is like a 5 and this is a 1. I say this because when I was in undergrad and stuff, I thought it was like EcoRV and like EcoRI. And then I probably embarrassed myself talking about them when they're actually numbers. And the numbers come from, and the name comes from like the bacteria that they come from. So the numbers like the one that they found in there. So well, 1 would be the first one they found in there. And then the name part is referring to the bacteria it was found in, and then like potentially the strain. So BAM-H1 is a common one that we use. And so it recognizes this sequence and cuts it in this place. The name just tells us it's that the first restriction enzyme discovered in this H strain of a bacteria called Bacillus amylox. I'm not going to try to say that, but this thingamabob. And so that's where the BAM comes from. And then the H is the strain, and then the 1 for the first enzyme described. You'll see that I have some text in today's presentation or today's post, and this is, you can find all of this on my blog. I have a lot more extensive stuff. So let's get into talking about how this interplays, the restriction enzymes that bacteria use interplay with the methyltransferases that they use. And so a methyl group is the CH3, and bacteria can transfer the CH3 group typically from like SAM, so S-adenosyl methionine, which we talked about in our post on methionine. And so it can transfer the CH3 group onto various things, different methyltransferases. And so bacteria have different methyltransferases that'll actually modify their own DNA to prevent them from getting cut up by their restriction enzymes. So basically what they do is these use code words too, so they recognize specific sequences and then they methylate them. So basically why I'm telling you about this isn't just because it's cool and because it's how the bacteria prevent chewing up their own DNA, but because it also comes into play when we're trying to use these enzymes in the lab. So the exact detail of this like RM system, so restriction modification system in different bacteria differs. And so sometimes they have the methyltransferase attached to a restriction enzyme, and sometimes they're separate. The ones that we use, the systems that have them separate are mostly useful for us because they can use their restriction enzymes part. But so typically a bacteria will have one of these complexes or these separate enzymes. Basically they have they'll recognize the same site. And so the restriction enzyme will cut the site if it's not methylated, but then it'll methylate it in its own DNA. And so because different bacteria have different sites that these complexes recognize, then we can use different bacteria and different strains of bacteria and different restriction enzymes from different bacteria. And so this is why we have such like a wide range of restriction enzymes to choose from, is because these bacteria each have like their own strategies kind of evolutionarily. They have adopted these sequences and that they recognize and protect in their own and cut in foreign DNA. So some of the methylases that they use are more specific and some of them are more generic. And this DAM one is one of the more generic ones. So what this one does is it recognizes this A and this GATC sequence, and it's going to methylate it. If it methylates it and it's not overlapping with a cut site that we want to use, we don't need to worry about this. But when they overlap, the methylation can hide the restriction enzymes code words. And so this is good for when the bacteria don't want to cut themselves, but it's bad for when we want to cut them. And so I'll talk more about there can be some uses where you need to be careful. You'll see things on the protocols, and that's in the restrictions for the enzymes that tell you about their compatibility. And then there are bacteria strains that you could use that would potentially like not have that methyltransferase. But why do we want this methyltransferase? And so DAM in particular, it has this function where what it's going to do is it's going to help the bacteria. It's going to help the bacteria like when they're replicating their DNA. And so basically what happens is that when they're copying their DNA, because DNA has two strands, these strands get unzipped, and then the DNA polymerase is going to go along these strands and it's going to copy each strand. It needs to know, it wants to know which strand is kind of like the template and which is the new one. And so there's this initially, the copies are going to be hemimethylated because the DNA polymerase, it can't add the methylation. The DAM adds the methylation. And so the DNA is going to get copied, and when it gets copied, it's originally going to be unmethylated. But the strand that the template strand that it was being copied from is going to be methylated. So initially you have the state that's hemimethylated. So one of the strands is methylated and the other strand isn't. This way, if there is an error, in one of these, then the fixer enzymes that come and try to correct the error know which strand was the actual strand and which strand was the errored strand. So they can recognize if there's a mismatch. And if there's a mismatch, then they can fix it, and they can fix it to be what would be complementary to the strand that was methylated, because that would be your parent strand. And so this is really helpful for bacteria, but it can get in the way if that DAM methylation is hiding one of your restriction sites. So most of the RNAs, most of the restriction enzymes we use in the lab don't overlap with these DAM sites, but some of them, such as CLA1, MBO1, and XBO1 might. There are a couple of other methyltransferases, though, that you might need to worry about. This other one that's pretty common is DCM. So DCM is another one that does like very widespread methylation, and it's going to methylate in this sequence, the second C here. And so W is just standing for an adenine or a thymidine, so just an A or a T. These sites aren't as common as the DAM sites. So the DAM sites are about 1 per 256 base pairs, whereas these are about 1 per 512. They can interfere with cutting by some of these. And you can find all the information about compatibility when you're looking up the different restriction enzymes. And so another one is EcoK1, and it can hide these cut sites. But because these sequences are longer and they're rarer, and you don't have to worry about them as much. So often you can avoid using these enzymes that are incompatible. If you can't, there are strains that are, say, like DAM and DCM minus, and so these are indicating that these don't have this DAM or DCM. But the reason why we wouldn't want to use these all the time is that then the DNA is more subject to having errors kept in, because the bacteria won't know which strand is going to be the strand to use. So there are some different types of methyltransferases that bacteria use. And so some of them recognize more specific sequences, and some of them are more generic, and some of them play a key role, like DAM plays a key role in helping the bacteria know which strand was which when they're replicating the DNA, to make sure that if there's any errors, they fix it based on the template strand, as opposed to based on the like the new strand that had the error introduced. Because DNA, because these methyltransferases, the methylation is only going to be, it's going to be added like in vivo. So in these bacteria cells, it's not going to be added if you are just like replicating DNA another way. So if you're using a PCR-based method to do cloning, say you have this plasmid and then you make copies of the sequence out of one plasmid and put it into another, such as with methods like SLIC, where you're basically, they're PCR-based methods in which you are making copies of a sequence from a parent vector. So if this vector was amplified in bacteria, so say you did like grew it up in bacteria and then did a mini prep and purified it out, that plasmid is going to be methylated. Unless you were using a bacteria strain that was lacking the methyltransferases, that is going to be methylated. But your insert is not, the thing you copied is not. And so if you don't purify out the thing you copied, you're still going to have the parent plasmid, and that parent plasmid may have the antibiotic resistance gene if it's like the same one that is used in the thing you're putting your sequence into. Then you can have parent plasmid carry over. So what we do is we use this other restriction enzyme, DPN1, and DPN1, it actually, as we've talked about methylation hiding restriction sites so that DNA don't cut themselves up, but DPN1 actually recognizes and cuts methylated DNA. It's a pretty weird one. So basically it comes from Streptococcus pneumoniae, and it's kind of switched roles with phages when it comes to methylation, potentially as part of like a sort of arms race between the phages and the bacteria. Some of the phages started methylating their own DNA, and so then the bacteria evolved so that this DPN1 only cuts methylated sites. And we could take advantage of this to chew up the methylated parent plasmid so that we only have our plasmid left over. Speaking of cloning and PCR methods, so often what we're doing is we're cutting a piece of DNA from one place and cutting a plasmid, so a circular piece of DNA that we can use to kind of like host our insert, so such as the instructions for making a protein, that would be like our insert. And we were trying to cut it out of one place and then put it in a plasmid so that we can grow it in bacteria. What's going to happen is that we can cut the plasmid that we want it in, and then we can cut the insert, and then we can stitch them together with a DNA ligase. When you do this, you want to use like the same restriction enzymes, or at least ones that generate complementary sticky ends. And typically what you do is you do this cutting, and then you actually run an agarose gel electrophoresis. So you're separating the DNA pieces by size, and then you do a gel purification where you're actually purifying out these pieces. Then you take the pieces you want and you mix them together, and then you stitch them together. But what if your sequence doesn't have the cut site like next to it? What you can actually do is you can use PCR to make copies of the insert, adding on these primers that have the restriction cut sites. And you can even do the same to make copies of the plasmid that have a cut site if the plasmid doesn't have the cut site that you want. And then you can use this to add on these cut sites and make these this cutting, and then you can stitch them together. Another option, if your DNA does not have a cut site, is that you can actually make like silent mutations that are going to introduce a cut site without changing the actual like protein instructions. So because of the redundancy of the genetic code, where these like 3-letter combinations, so codons spell like specific amino acids. And so the DNA gets copied to mRNA, gets made into protein based on these instructions, but you can have multiple words spell the same thing. And so if you can take a site that isn't a site for a cutter and you can turn it into a site for a cutter. You can also do the opposite. You can change the sequence in a way that's not going to change the protein, introduce the silent site, but you can actually remove a site, because sometimes if you imagine that when you're doing this, you want to make sure that you only have like a single cut site if your goal is to be cloning, or you have a cut site on either side for the same enzyme. But you need to make sure that you're not having cut sites where you don't want them. And so there are different. Some of the restriction enzymes, if they have more promiscuous sequences, whereas others have more highly specific, you can think of these as kind of like passwords. The longer it is, the less likely it is to happen randomly. And so if some of these have longer sequences that they recognize, some have a little promiscuity in the sequence that they recognize. So it might be like, oh, you can have an A or a T here, or that sort of thing. Or often they have like an N there in the sequence. When you see the sequence that it cuts, it'll have an N that's representing like it could be any nucleotide. So those can be problematic when you're trying to do some restriction cloning, but they can be helpful when you want to have more cuts. So there's software that you can use, and I'll show you that you can actually see how many sites there are and get a list of enzymes that like cut in a single place in a various plasmid. So why might you want to do this? In addition to cloning, you can also use these enzyme cut sites in order to get information about things like genetics. So basically, there's this thing called restriction fragment length polymorphism, which is this idea that there are restriction sites, recognition sites in our DNA because these sequences just happen randomly. And we haven't like evolved in this presence of these like, we haven't evolved to avoid using these sequences like bacteria have. So these sequences are going to occur in our DNA. But the sequences between these cut sites can vary between people. We have a lot of sequences in our DNA that are like repetitive, and people can have different numbers of these repetitive sequences. And so there's going to be different lengths if you were between the cut sites. And so people can have different lengths, and then this can be used for like genetic tracing and for like crime scene analysis. These days, typically instead of looking for these restriction lengths, people use like PCR and use like primers instead of restriction sites. They'll make copies of those sequences and then see how long they are. We do a similar thing with analytical restriction enzyme digest, which we can use after we do cloning to kind of get an idea whether our cloning actually worked. So there are different ways that you can do this. If you have a cut site in your plasmid, or you have a cut site outside of your, in your insert, or you have it outside of your insert, the key thing is that you will have different size pieces depending on whether or not your cloning was successful. And then you can use this to introduce these cuts and then see how big the pieces are and if they would match what you would expect. So here is a case where you might want to introduce a silent cut site or remove a silent cut site in order to facilitate the analysis. And it's also important, though, to note that this is not going to tell you if the whole sequence is correct, just if the size looks like it might be right. And then what you want to do is you want to really check that the cloning is actually successful. What you're going to want to do is you're going to send it for sequencing in order to verify that the whole sequence is actually correct and that you don't have any typos introduced. Speaking of controls, when you're doing restriction cloning, you also want to do some controls to make sure that the parts that you do after you actually do the cloning worked. And so typically these plasmids have an antibiotic resistance gene that you're using to select for bacteria that actually took in your plasmid. You want to use an uncut vector as a positive control that this transformation actually works. So you actually got this into the cells, and these cells are competent, so they were able to actually take in the DNA. So each of these globby dots is going to be a colony, which should correspond to genetically identical bacteria, and only the ones that have the plasmid should be able to grow. So this is going to be your positive control for these competency of these cells. And then you want a cut vector that's going to serve as a negative control that's going to tell you that all the vector got cut. So you didn't have any like parent vectors or self-circularized vectors. And so this is also going to show that your antibiotics were effective. And this is going to, so when you're doing that gel purification, that you actually purified out the parent, and you're not just looking at that. Okay, so let's look a little more at how you actually go about doing this sort of thing in practice when you're designing some sort of using a restriction enzyme. So as I mentioned, NEB is kind of like the monopoly company, I guess, in this game, the restriction enzyme game, and they have a ton of restriction enzymes. And so you can see a lot of these have ends, which means that these sequences, they're more promiscuous. You can also see that some of them are actually like cutting outside of the sequence that they recognize. There are different types of restriction enzymes, basically. You'll also see a lot of information over here about various properties. So like incubation temperature, the buffer, we'll talk more about the buffers, whether they're heat inactivable, whether they're sensitive to like various methylations, that sort of thing. So this is just a general enzyme finder, and you can search for specific ones, or you can actually, they have a bunch of tools like NEB Cutter where you can actually, it'll find restriction sites for you. There used to be a site called WhatCut. I haven't been able to reach it, but I found this other site, Silent Mutator, where these are going to find mutations that you can make in order to introduce or eliminate a cut site for when you're trying to do some sort of cloning or some sort of analysis, as we talked about. So let's look at a specific example. Let's look at BAMH1, which is the one that I had mentioned earlier. And so you can see it'll show you the sequence and the place where it cuts with these arrows. And so you can see that it has this palindromic sequence and it's cutting in this place. In terms of like the product information, so it's going to basically tell you the concentration is going to be given in units per mil. And I've talked about enzyme activity units. And so basically enzyme activity units are basically whatever they say it is. And so as long as you define the unit, then it can, that's giving you an idea about how active the enzyme is. And so here, if you go to properties and usage, you can see that one unit is defined as the amount required to digest this, blah, blah, blah. And then it's also going to tell you about the activity in various buffers. And so the buffer information gets kind of confusing because the buffers have changed over the years. So it used to be that you would have a buffer that was like used with buffer 3. But with buffer 3, you would have to add BSA to it. So BSA, bovine serum albumin, is this abundant protein from like cow bovine blood, and it's basically, it's kind of this generic protein that we use for a lot of different things. But in this case, it's being used to prevent absorption to the sides of the tube of your stuff. And so basically, you can think of the BSA as kind of, if you have things like, if you have a bunch of stuff in the tube, and some of that stuff is going to stick to the sides of the tube because some stuff always sticks to the sides of the tube. You want that stuff that's sticking to be the BSA and not your stuff. You want it to be something other than your stuff. And so if you put a bunch of BSA in there, now your stuff is less likely to stick. I've also seen some other potential things with the BSA kind of binding to, and like proteases and other stuff, you don't want them to have weird activity on various things, and just kind of keeping things happy. But so it used to be that you would have a buffer like 3, and then you would add BSA to it. Then they introduced buffers with the BSA already in them, and so this would be like 3.1. So there were several different buffers. So some of your enzymes would come with buffer 1, some of them would come with buffer 2, some of them would come with buffer 3. And so then this could be confusing, and this could be difficult if you were trying to do like a double digest. So when you're doing one of those clonings and you want to use 2 different enzymes at the same time, so that you can cut 2 different sites, and then you like cut on with different enzymes on each side and then stitch them together. If you have a if they had really different buffer compatibilities, you would have to do those reactions separately. But if they have similar activity in the same buffer, then you could do it together. And so NEB introduced this like cut smart buffer that has better compatibility for most of the enzymes. Like most of their enzymes will work in this buffer. And you can see that it'll also give you the activity in different types of buffers. So now you might be wondering about this R. So this R is this recombinant. So basically they used to use BSA, and that was like purified from like the cow. but now, then you have like batch to batch variability, and you could have various issues. And so, and also like medical grade stuff, I'm assuming. But basically, then they started making their own like recombinant albumin. So basically, recombinant, like we've been talking about, is where you recombine pieces of DNA. And when you do that, you can put in the instructions for making this albumin protein and then make it. And so then you have this non-animal source, and you have the better purity and less variability and that sort of thing. So then they changed their buffers, and so now the ones with the R, it has this recombinant albumin instead of the traditional BSA. So that's what the difference is there, but you should still be able to use the old one without problems. Speaking of using old ones, like these enzymes have like expiration dates, but I have used restriction enzymes that are so old without a problem. I don't know if I would have a little better activity with the newer one, probably. And if you're having issues, you should definitely test and see if your thing is still active with some positive controls, but don't just throw it out because you're past the expiration date. You also see it'll tell you about methylation sensitivity. So this one is not sensitive to the methylation, so you don't need to worry about that. So those are some various things to look at when you are planning out some sort of restriction enzyme digest, and they also have information about how to use these. So typical reactions is super duper simple. The buffer, when it comes, it's like a 10 times concentrate. So if you were doing a 50 microliter reaction, you would have 5 microliters of that. The incubation time can vary for these different enzymes. They have some where they say you can decrease to 5 to 15 minutes if it's like a time saver qualified one. I'm not exactly sure, but you might have to optimize various things with your, depending on how many sites are on your DNA rather than like how much mgs of your DNA you have or whatever. Because remember, it's actually the sites that's actually mattering, not the total like weight of your DNA. So more on that in my rants on enzyme activity and specific activity and various stuff. So some of these you actually can terminate if you want to make sure that it stops cutting. Some are heat inactivatable, or you can remove the enzyme using some sort of spin column that's going to, because the enzyme is big, it's going to get trapped in the column, and then the other stuff is going to come through. In terms of storage, we typically keep them in those like little storage blocks in the freezer. And they have glycerol in them, so they're not going to look like frozen solid. They're going to be like jelly. But you need to keep them like in -20 and handle them with care. And that's the basics with working with restriction enzymes. And they have a lot of tools to help you. And again, I don't work for NEB, but they do have a lot of good resources. So that's it for today, and good luck with all of your experiments.